And very quickly, just show that yes, treatment does make a difference. This is look at triclosan, that antibacterial ingredient that's all in your soaps and so forth. Got 3 plants, 3 are activated sludge, and you got your trickling filter, and the trickling filter is a lot less efficient in reducing concentration. Not saying removal. Removal is very tricky terminology, because we're not saying it's being removed, because most of the general public would think removal that's breaking down and gone. Removal could simply be degrading to a new compound, a transformation product, or partitioning out of the water into the sludge. So really want to make sure that this is a reduction, not a removal, and show that technology, the treatment technology, does make a difference. This is what I kind of alluded to earlier in terms of this happens to be a music festival in Taiwan, where it's actually a massive music. This was that like the city of Detroit? Is that basically? Certainly, it's bigger than it's 3 times the size of the city of Des Moines over this week-long time period. And basically, want to focus on MDMA, which is ecstasy. And you can see during the music festival, it increased dramatically and fell off after all the music people left. So again, it just shows, and I think that same could be true whether you got a big sporting event or whatever. You got people coming in and can not only, and it could be more typical things like caffeine or just whatever. In this case, happens to be some recreational stuff going on, but just shows that it can have an impact on what's coming out of your wastewater treatment plant. And so same as on the wastewater side is on the drinking water side. These compounds were not part of the equation when these plants were built, so it's not surprising that things will blow through either unremoved or partially removed on the drinking water side as well, as I alluded to earlier. I think this is, in my mind, one of the earlier examples of looking at a drinking water treatment plant. This is in New Jersey, so there's heavily populated, heavy population density, lots of potential sources. There's wastewater treatment plants upstream of this drinking water treatment plant intake. You can see there's lots of stuff from pharmaceuticals and personal care products and a whole range of things. And when you look at the source water, again, or the finished water, there's some removal, some reduction. Sorry, I don't want to use that term removal. But there is some improvement, but it varies on the chemical itself. Some really had no change in the percent detections. Others were a little bit better, but it does show that there can be, if something in your source water, there can be things that end up in your finished water. And so we followed up. There's some other work that Bonatti and others had done. I think they were in the Nevada Surface Water Authority, I think, but did a nice study. Looked at more plants, looked at like 18 plants, but their target compounds were limited, more in the 20, 30 pharmaceuticals. So working with EPA, EPA is very interested in determining what compounds need to go on their unregulated, their UCMLR, which is kind of the precursor to ultimately creating drinking water standards. So we only did 20 on only. It's certainly more than Bonatti did, but it's not a huge number. Paired source and treated drinking water, but really through the kitchen sink at measuring what we can measure in these compounds, both chemical and microbial and analytes. Really went overboard, I think. Not overboard, but extensive, more than what anybody else does in terms of looking at QA/QC. 70% of the analyses were QA/QC samples. So we want to make sure that when we said something was in the drinking water, we knew it was there, not just because of analytical variability or whatever. I'm not going to get into these other aspects because of time. But these are the compounds that were most commonly detected in source water. So they're in at least 7 of the 25 source water plants. Again, whole range of things from pharmaceuticals to perfluorinated compounds, kind of a grab bag of other anthropogenic wastewater indicators, inorganics and microorganisms. And when you flip to, again, not surprised, kind of just eyeball that trend there, and we go to finished water, you can see big differences in the number of pharmaceuticals dropped. Again, these are those that were in at least 7 of the 25 plants. All the microorganisms fall off, but really the PFCs in particular and the inorganics really had very little change. So the treatment has little impact on perfluorinateds or the inorganics. Okay, so kind of keep moving on. We doing okay for time? All right. And just, the big question everybody wants to know is, well, what are the consequences from the presence of these compounds? And certainly, no question, there's environmental uptake. And that's from a range of plant and animal tissue. That's from simple things like, corn and lettuce, potatoes, but these are things that humans consume. So there is uptake if you have things in your, whether it's your hog manure or your biosolids get applied to land surface, and you're using that as a source of fertilizer for your plants, they will uptake it, and you could, it could be in the food you consume. Is that a big pathway? That's another question. It's in earthworms, mussels, so basically it's in a range of things is from bacteria up to higher organisms like dolphins and certainly probably humans. We all test our blood. We're going to have fragrances and all kinds of compounds in there as well. But I think in terms of understanding effects, in my mind, again, I'm not a toxicologist, so I may be a little on a limb here, but I consider there's real limitations to a lot of the ongoing toxicological research that's being done, partly because I think the standardized tests really underestimate effects. And this paper here that was published just this year, they looked at 4 pharmaceuticals and they found them individually were not toxic, even when they smacked them up to milligram per liter levels using the standardized acute tests, didn't find any effects. However, when we looked at these same compounds and looked at these embryo larval development chronic tests and whacked them at environmentally realistic concentrations, all had effects shown. So it really shows that these chronic behavioral effects, those are what's going on out there. So I think the standardized tests are really underestimating really what's potential risk that's what's going on. And we've talked about this numerous times since I've been here, this whole chemical mixture issue where most of the tests consider one compound at a time. Well, that's not reality. When you go to a stream where a fish is living in or a worm, soil that a worm is in, it is a whole cocktail, a whole mixture of types of things. And certainly this paper here that was published last year showing that there's mixture effects even when each individual component is present at low levels and not determined to have an individual effect. When you throw them together and consider them as a mixture, there is effects going on. So that's a real challenge, just to understand not just things on an individual compound basis, but as a mixture. But even given those limitations, there's mounting evidence that there's ecological effects. And these are just a few. This is growing. There's all kinds of whether it's irreversible fish masculinization or decreased denitrification rates when exposed to antibiotics and so forth. So there's a whole range of things. Still, there's less to know about the complex mixture issues, but given it shows that there can be. Not every CEC is going to have an effect, but there's a number of those that are shown to definitely have environmental impacts. Kind of the extreme acute example is with the vultures in the India-Pakistan region, where just in a matter of 3 years, the population was almost decimated, where there was millions of vultures at one point in time, and all this coincided with the use of diclofenac. This is a non-steroidal anti-inflammatory used to treat cattle, and in some cases the cattle just don't survive. And in that part of the world, the disposal mechanism of the carcasses is let the vultures eat them. It's a very, very efficient way to handle it. And it just happens to be these vultures were very vulnerable to diclofenac, caused renal failure, just was unable to excrete the diclofenac, and it really caused the population to collapse. So this to me is a classic example of you can do every test for toxicity that are known to man, and you still have unintended consequences. You just can't predict everything that could go on when you expose things to a whole range of organisms just may be way more sensitive than you ever would have imagined. And I want to kind of wrap up here with what I consider one of our research laboratories where we do hypothesis testing in the real world. This happens in Boulder Creek in Colorado, and I just want to show this progression of how we've taken, we build upon the previous knowledge of what we gained before to develop new hypothesis and new understanding. Where this was some work that with our colleagues at the University of Colorado, they're looking at Boulder Creek. On this stretch was above and below the wastewater treatment plant. Above the plant, it fortuitously had a low head dam, so it separated the native fish populations. In this case, it was white sucker, and they were showing below the plant where only the fish below were having intersex. There were skewed female to male sex ratios below the plant where it's more of a 50-50. Above, decreased sperm abundance, elevated plasma vitellogenin. Vitellogenin is a female egg yolk protein, so the males should not have elevated levels. So all those are showing signs that there's a population level endocrine disruption going on in the system. And as it turned out, this is before we knew each other. We're working in the same stream, and now we're working together. We're looking at our chemical characterization of the same site above, and this is a range of estrogenic compounds above the plant. Now this is basically mountain stream water that's coming down the mountains, the Front Range. No estrogenicity. This is a pure effluent. You see, not only is it high estrogenicity, but it's not a single compound. It's a mixture of compounds that are causing this estrogenicity. And we go below the plant, and this reduction is basically dilution. So it's not attenuation. It's essentially dilution with this low concentrations above water. So really the hypothesis, we're seeing differences in native fish populations. We're seeing differences in chemistry above and below the wastewater treatment plant. So the obvious hypothesis is it the plant that's causing the effects to the fish. So I tip my hat to the city of Boulder. They're very gracious. They allowed us on site to use these mobile exposure labs. And so rather than taking barrels of effluent and bringing it into laboratories, that's like a snapshot in time, bring it to the laboratory. But as we all know, as that barrel sits there, no matter how well you keep it chilled or preserved, things are going to start happening to those concentrations. So this basically you're taking the laboratory into the field and you're doing in situ field experiments. So they're continually pumping water both from the effluent itself and from the upstream of the plant itself. So you have your control water and your source water simultaneously in the same location. They're pumped in two different tanks, and then they're brought to temperature equilibrium, because hormone levels, if you got warm effluent and cold stream water, can affect hormone levels within these male fatty minnows that we're looking at. So bottom line is we're doing control experiments, and we can do a whole range from basically 100% reference water or 0% effluent to 100% effluent. And in this case, we also have a 50-50 mix, but we could do any sorts of combination in between. These are our male fathead minnows. They have these secondary sex characteristics of these nuptial tubercles, these bumps on the head, so you can externally differentiate a male from female. And you can see very quickly within 7 days when exposure to these, even at 50-50 mix, you're getting feminization. You're basically starting to look like males or starting to look like females. So we're getting a reduction of these nuptial tubercles. They're looking like males or looking like females or acting like females. So that led us, so after we realized that, published that paper, the plant itself, not because of this particular study, but they went through a huge upgrade, like a $50 million upgrade from a trickling filter, which I showed previously was not good at reducing a lot of concentrations being released in the effluent, to an activated sludge, which is a much more efficient mechanism. So that led us to, so if they're changing this technology, and we know that can have an effect, that gave us another line of hypothesis testing. Will that have an impact on the estrogenicity of the effluent that's being released to Boulder Creek? So we kept these mobile exposure labs and experiments rolling, and we found that there in fact was a significant decrease in the estrogenicity of effluent. So very estrogenic before the upgrade, no estrogenicity after the upgrade. And so we repeated our experiments. Again, we saw that there was greatly reduced endocrine disruption relative to pre-upgrade conditions. So that's great news. So we're not seeing that feminization of the male fatheads. So that led us to say, okay, if now the plant is improving, it's not releasing those compounds into the stream, will that ultimately have then an impact to those white sucker that we showed previously that were feminized and skewed sex ratios and intersex fish and so forth? Now, white sucker are fairly long-lived, so this took place in 2007. You can't the next year immediately do another fish community structure survey. You have to wait a bit for basically the next generation to come along. So we waited 4 years, and in fact, we did see a huge recovery in the endocrine disruption of the wild fish. The intersex we didn't find after the upgrade. The skewed sex ratios came back to more of a 50-50 or close to a 50-50 ratio. Vitellogenin was greatly reduced. So again, that's good news. That really shows that with these kind of field laboratories, you can keep building on these hypotheses as you go from each study and building on from your previous studies. And really, that's this approach that we use now, at least we're trying to use more and more, as when we're looking at a system, whether it's a river or whatever, we're still doing our analytical methods. So as best we can, we're characterizing that, but that's very expensive. And also, you can never measure everything, no matter how hard we do. Even when you're measuring 700 compounds, there's tens of thousands, if not hundreds of thousands of possibilities, particularly when you consider all the transformation of the products these active ingredients can degrade into. So that's where we get into then looking at these bioassays for biological activity. Is the water estrogenic? You may be measuring hormones, but you're not measuring every possible hormone or every possible estrogen mimicker. So you look at basically total estrogenicity and thyroid activity. So it again gives us a screening of activity. But then we also do these mobile labs, like I just talked about at Boulder Creek, both of kind of taking test species and exposing fish to test species. Then we have their wild fish screening as well. So all these, we're trying to do an integrated approach, all have their strengths, and try to do these all simultaneously so we can have the most comprehensive understanding of environmental effects as possible. So I'll end here with, is there human health effects? Obviously, this is $1 million question, but it's really difficult. Now I'm kind of outside my area of expertise, but certainly, it's one thing to expose a fathead minnow to effluent, but we can't, take college students and say, well, drink this effluent and we'll see what happens to you. That's just, it's not something we can do, at least not something we should be doing. So these certainly difficulty in conducting these epidemiological studies, but obviously there's concern for fetal exposure. The diethyl stilbestrol, this is something that was given to pregnant women back in the 60s or 70s, I'm trying to remember, and definitely found that had an impact to the unborn fetus. So that, there's certainly that type of exposure is known to be an issue. Animals and sentinels, like I mentioned previously, we know these compounds can have an effect to both terrestrial and aquatic organisms. If there's effects to animals, should we be concerned about having potential effects to humans? Certainly, as I mentioned, there's definitely pharmaceuticals in drinking water. There is that kind of two camps there, where some just think we shouldn't have any exposure to trace levels in our drinking water is bad, and the other camp says, well, it takes water towers of you have to drink a water tower a day to get a single exposure, and say, well, that's an acceptable risk. It takes so much water to get a single dose that's an acceptable risk. I'm kind of in the middle, where I think it's good news that generally the concentrations we're seeing, particularly of pharmaceuticals in drinking water, is low, but I think we really need more research to know, is there unintended consequences from this long-term chronic exposure levels. And again, part of that is the epigenetics, early exposure could potentially lead to late effects. This transgenerational transmission, where chemical exposure now could impact future generations. And certainly, again, going back to the animal example, there's definitely been shown that effects can be passed to the F2 and F3 generation even after no further exposures has been occurred. So again, it's not to say that will happen in humans, but if it can happen in animals, it's something we need to at least consider, because that's really, I think, a long-term question is, what are we doing? Obviously, we're living longer. Pharmaceuticals and others have their place, but we need to make sure that we are doing our due diligence to make sure that we are not passing risk to future generations. And with that, I know I seem like I talk fast, but I'll certainly field any questions, if there is any.